You can start.
Good morning. Today we are gathering here for a special issue, that's safety. So in this regard, I am Dr. Satisha, scientist in ICR SIBA. Welcome you all. To beginning, we have here the chief guest, Dr. Tirumurugan, and our director, Dr. Kuldeep Lal, online, and acting director, Dr. KP Jitendran, and also the chairman, Dr. Shishekar. So to begin the program, I request Dr. Makesh to welcome the gathering and also give a brief introduction about the program.
A very good morning to all. At the outset, I would like to welcome our director online, Dr. Kuldeep Lal, and also the director in charge, Dr. Jitendran, and our special invitee, Dr. KG Tirumurugan, who is the project director at Translational Research Center on Veterinary Biologicals, for this important event where he'll be delivering the guest lecture. I request our director to welcome our chief guest with a floral bouquet. I also welcome the chairman, Institutional Biosafety Committee, Dr. Shishekar, heads of the divisions, senior colleagues, friends, and scholars. So this talk actually it's to create an awareness among us to handle biosafety issues. Biosafety is a very important aspect. We all know that the corona has taught us a lot of lessons, how a single virus can turn the world upside down. It is, of course, there's a lot of speculation how about the origin of the virus, but imagine a virus or a pathogen getting out of a lab, which can cause this amount of death or millions of people dying. Of course, it is not only the case with human pathogen. In a veterinary animal or a plant or a fish pathogen can equally be devastating, provided, suppose in case if a wrong pathogen, virulent pathogen or a transgenic organism or a transgenic species or a recombinant organism, if it escapes the lab, we do not know what type of impact it can cause in the environment. So it is our duty to prevent such escapes into the environment, to prevent any untoward events, because we do not know how, unless the organism is proven that it is not going to harm the environment or the human being or the other living organisms, we cannot take chances, we cannot let them go into the environment. So we have to have containment measures to see that these organisms by mistake also do not go into the environment. And that is why we have this Institutional Biosafety Committee. Of course, this committee is there in almost all R&D organizations have handling virulent organisms or recombinant organisms or transgenic species. So we also have an IBSC committee where we take care of approving any research work, all research work which incorporates virulent organisms or recombinant or transgenic species has to be approved by the Institutional Biosafety Committee. And of course, if large scale means it has to be approved by the apex body, that is the RCGM, Review Committee on Genetic Manipulations. So this is a very well thought process has been established to prevent such outbreak or leakage of virulent organisms. And here, Dr. Tirumurugan, he is also our biosafety external expert for our Institutional Biosafety Committee, and he also happens to be the member of RCGM, the Review Committee on Genetic Manipulation under the Government of Department of Biotechnology, Government of India. And we in fact take his advice whenever we want to get some approval from RCGM also. So he's very kind enough to cooperate in that aspect. So here today he's going to tell us about what is the essential, what are the projects which needs to be approved by the IBSC, what are the process in getting your project, suppose if it involves a virulent pathogen or a recombinant organism, how to get it approved from IBSC or RCGM, and what are the things you have to do in your lab to prevent this escape of these potential organisms to the environment, which can potentially infect other organisms or human beings. So we are very eager to listen from you, Dr. Tirumurugan. So we hope you will be enlightening us, because it's a very important event. Many of us are not aware of this process, and also some of the process which we need to handle at our laboratory. So with this, I welcome you all to this very important lecture, and thank you.
Thank you, sir. Yeah, this biosafety committee, it is a process, but as sir has told, post-corona, the importance of this committee, it has become more internationally important. So now I request the chairman of our committee, Dr. Shashikar, to give his address.
Good morning, respected director, Dr. Tirumurugan. Welcome to our institute, Dr. Jitendran, and dear colleagues. So as a chairman of this committee, we are I think we are making a good initiation by calling Dr. Tirumurugan and to give an aspect or overview of the biosafety protocols which needs to be followed. Now, as Makesh was saying, that he is going to cover on large aspects of his talk on how the protocols has to be followed. Mainly, I feel there are two aspects into this. One is the escape of pathogens. So that's another biosafety. I think the majorly is going to focus on that. But I feel there's another aspect where escape of pathogens is always there, like how to protect the environment and the other organisms. But the protection to the self in the laboratory is also equally important. And that also has to be taken care. I think I and Makesh need to plan into that kind of talks also, where the students, we need to make them more aware how to first work in the lab, what are the safety protocols to be followed. And I know everyone uses the gloves, but where not to use the gloves is also equally important. So I have brought that to the notice, and some awareness has to be built, because just to give an example, because we go with an apron or a lab or our protective gear with gloves in the lab, and that should be left in the lab while coming out of the premises. So coming out to the open common areas, wearing apron and gloves is also prohibited. So those are very basic things which we need to understand. And in my experience, I think I have gone to 2-3 foreign countries for a short training program. But before I was allowed to enter to the lab, I was made to undergo a 1 week short course. And they were all the new entrants into the lab were forced to undergo that short course, pass the exam, and there they used to tell what are the protocols need to be followed in lab. So that includes how to dispose of the sharp nails and how to wear the mask, what are the dangerous chemicals, how to store that. So that is also equally important. Anyway, I'm not going to take much time. And once again, I welcome you, Dr. Tirumurugan. Thank you very much.
Thank you, sir. Even when we walk through or passing through some medical college, we will be seeing several aspirant doctors walking with the lab coat or apron. So it is supposed to be where in the clinic, not on the roadside to show whether they are doctors. So similarly here also, when we are walking between the labs, lab coat is for our personal safety. It's not for showing that we are working. Anyway, on a lighter note. Okay. So I request now that Dr. K.P. Jitendran, Director in charge here, for his address.
Today's chief guest and the speaker, Dr. K.G. Tirumurugan, he is the project director, translation project on veterinary biologists at Talibas. And director is online, I think, yeah. And chairman, Institute Biosafety Committee, then Dr. Magesh is the member secretary of IBSE, and all my colleagues, scientists in charges, and students. In fact, this talk is arranged with a lot of vision. Of course, even 2 weeks back, I was just discussing the same point in my division meeting also. One of the most important things which I wanted to highlight here, that's all spoken by Dr. Shekhar already, but still, I just wanted to highlight a few points. One is that this animal ethics and biosafety. This is one of the two things which we have to give a lot of importance in our institute. And unfortunately, this is being neglected to a large extent. One of the reasons for this negligence is that most of the pathogens which we are handling in our institute, whether it is shrimp or fish, there's not infectious to human beings. So we started taking some relaxation on that account and started handling the things very not seriously. That is one thing. And another thing is that there are few bacteria species which is pathogenic to human beings. Otherwise, most of the virus is not infectious to human beings, and parasites also. A large majority of the parasites, except a few tapeworms, is not infectious to human beings. And similarly, there are some area which we traditionally neglect in fisheries field. That is zoonotic diseases. Of course, I think even 2 months back or 1.5 months back, there was a meeting conducted by Tamil Nadu Fisheries Department. From our institute, Dr. Anandaraja attended the meeting. And there was a lot of discussion about the zoonotic disease, which is passing on to the community workers in the field. And they asked us to do some work on that. Then I think he discussed all the issues and what are the issues. Even there is no agency to fund that type of work going on. And they also requested us to do that work, but without any funding. That is the importance which we are giving for this type of work, because they want the work to be done, but no funding. And unfortunately, to my knowledge, there is not even a single project operating on zoonotic disease in any of the fisheries. That is hard fact only. There's no doubt about that. And at the same time, we talk in almost all the conference, you will find at least one session on One Health approach. And whether it is animal, human beings, or even any other environment issues. And this is also gaining a lot of importance, but there is all restricted only in conferences and symposiums, not in much in practice. That is one thing. And today's lecture, I'm sure that the biggest beneficiary will be the students. Problem Dr. Shekhar already highlighted. One of the issue which I found is that most of the students are, as of now, is freshers. Normally, all these students learn their laboratory techniques, not from the book. They learn basically from their senior lot. How to even start a postmortem, or even how to start a dissection. They will be where to cut first, that type of things will be learning from the seniors. And here what happened is that there was a COVID period of 1.5 years. There's seniors and everything, they went out of the institute, and the lot which is present here, they're all invariably, 90% of them are new lot, and they don't have that luxury of having this experience in their hand, and they started doing all this. So it is our responsibility and job to empower with them with the good laboratory practice and good animal ethics practice also in the institute. I hope that this part, Dr. Thirumurugan will be handling it nicely, and all the best to the speaker, and thank you so much.
Thank you, sir. Today, our director, Dr. Kuldeep Lal, sir, though he is on official tour, he has joined us online. So I request sir to give some remark on this biosafety.
Good morning, and thank you, Satish. Dr. Jitendran, Dr. Thirumurugan, Dr. Shekhar, and Dr. Makesh. It's nice to see that this initiative being taken to create awareness of laboratory procedures. It has got many aspects, and many of these previous speakers have spoken on this. One is the most concerning is the pathogen, their spread, their escape, and how to handle. And definitely the safety of the workers who are using that. They should be well aware of about how to handle any particular equipment, or while they are handling some chemicals, how to go about their disposal, or how they means at that time what care they have to take in that they are not to touch some surfaces or all that. And as Shekhar told that use of gloves also has to be judicious. It's very important. Most of the time when we enter the lab, we try to remove the shoes where we don't go. But we also have to think that sweat of our the foot can also create the contamination among in the laboratory. We have to be very overall. We have to be concerned, and at the same time, we need to have standard operating procedures for each lab. Another aspect is, though at present genetically modified organisms are not widely accepted within India at the policy level, but many of the time for the research purpose, we use the genetically modified cells or we modify the cells. Now that also falls under the purview of the biosafety, because that can have the environmental concern. Zoonotic is one concern, but zoonotic has not been much relevant while we are taking the fisheries research, because at present, as far as I know, not much of the pathogens which are of the interest in the shrimp or fish, they have been having any much of the zoonotic importance. But at the same time, the surfaces which comes, they can carry the pathogens of zoonotic interest. So we have to look overall the laboratory, human, and in general environmental health when we look towards this. Maybe I will expect not from the today's talk. I am thankful to Dr. Tirumurugan who accepted our invitation to share his experience and the knowledge, because this itself is a area of science, having the biosafety protocols placed in a laboratory. And from this talk, I'll expect our people, we can have make a good start from here and make our consultation and make appropriate changes in our laboratory handling procedures, and try to make aware the people who come for our projects to how to have those safety procedures, at least for their own sake. I know some of the cases where the hazard happened. We must be teaching our students very well that when to use micropipette or when to use the glass pipettes. It's a one of the small thing, but it can create hazard. So they are not substitute to each other. Thank you very much. I again thank Dr. Tirumurugan and our committee and Dr. Makesh for organizing this. Thank you very much. I look forward to learn more from this. Thank you.
Thank you, sir. Yes, of course, biosafety is not a responsibility of a committee or it has to be responsibility of an individual, as their own professional attitude it has to be every day, every night, or it is like a 24/7. So continuing the program, I request Dr. Tirumurugan to give the most expected waiting presentation. But before that, for the audience, I want to just briefly introduce our chief guest about his background and his professional achievements. So Dr. K.G. Tirumurugan, he is professor and project director in charge of Translational Research Platform for Veterinary Biologicals in Tanvas. And he is a member in review committee of genetic manipulation of DBT. And also he is a member in interministerial committee for certification of BSL 3 facilities of DBT. He is basically a virologist with specialization in animal biotechnology from Madras Veterinary College of Tanwas, and his research area include innate immunity, signal transduction, and development of diagnostics for diseases of livestock importance. He had a postdoc associateship at the University of Minnesota, USA, to understand the contribution of CD38 and its downstream signaling to calcium ion mobilization in smooth muscles and its role in airway hyperresponsiveness. He is a recipient of a travel grant from FAO, United Nations, and OIE to attend the PPR Grand meeting at International Livestock Research Institute, Nairobi, for his research work on PPRV in unnatural host. He received several awards for his contribution to veterinary research in India and also abroad. He has 89 publications with 954 citations with the H index of 15 and I10 index of 22 since past 5 years. He has 1 patent granted, 2 patents filed, and also commercialized his technologies for betterment of the sector. He is currently heading the translation research platform of Veterinary Biologicals, a unique platform program between DBT and Tanwas, which operates intending to convert science to commercializable technologies by fostering an inclusive ecosystem involving academic, industry, and regulatory agencies, a huge responsibility. He is also duly supported by externally funded grants from several agencies obtained through competitive mode. He is a member of national level committees with specific reference to biosafety and rDNA research. So this is a brief information, though the list goes very long, it will be consuming a lot of time. So I request with all his professional experience to enrich us on biosafety in laboratories. Yes. And they are so,
Good morning to the learned audience here, respected director online and the director in charge here, and the learned colleagues of this institute, the young researchers. Probably like a lot of things have been told by the officials here. It's not like a policing job that happens in an institute, actually. So biosafety is not a kind of a policing work. It's a kind of a situation where you follow some research ethics. So ethics to protect yourself, ethics to the lab, and ethics to the institute. Finally ending up with something which is going to be supported both to the environment too. So it's only a kind of an ethics rather than a policing job. So I've just put a couple of things into this presentation, not only to provide some basics into what has to be followed in a lab. Let me also go through some in terms of what we actually practice in the lab rather than putting some slides. As a person belonging to a research fraternity, it is very critical that you have to know what is happening in terms of regulations, because like regulations is one which we lack completely. Example, to say in terms of regulations pertaining to biosafety, RCGM is one. It has a committee members of 26. And if you look into the committee profile, not much of the people get into it because like there's lack of regulatory knowledge. And why products don't get up into the market, a lot of research is being done in the institute. Not much gets diverted into a market. It results in a kind of a technology transfer to an institute or a company, but beyond that, it doesn't go back. So that's because like we lack a lot of things in terms of regulations pertaining to what is required in an industry before you start your work also. So regulation is one where a lot of people lack awareness, not only in terms of trying to understand it. Whenever it comes to regulation, people look at a lot of data, a lot of acts, rules, guidelines. So if you try to look into a simplified way of approaching that, it's going to be easy for people to understand. So I just started my position as a member secretary like Magesh, if I'm right, like 2012. So 2012, so 7 years as a DBT nominee, and then across different institutes, and had an option to enter into RCGM in 2019. So it's not like a kind of a scenario where it's difficult for everybody. It's a process which people have to take it up into mind, try to look at what is actually present now. And people also have to get updated. Like in science, we try to update ourselves. What is happening? So you look at new techniques, implementing them. The same way, there are a lot of revisions which happens also. So which also have to be taken care. So quickly, I've just put it, biosafety research, investigator to the regulatory pathway, which means like you trying to understand what is the basic there, how the regulatory agency puts a couple of requirements, where are the places where you can access these things, and what can be done effectively. Okay. So I just wanted to start this lecture with a small caution, putting out a small video. Okay, just go through. Probably like this would also have been come through a WhatsApp in terms of sharing. And you didn't put the entire folder. I'll just pay. Either share one of the. Will it go online? Just check this video alone. So the critical point here, you do a lot of things. How to just take care. So you do a lot of things in terms of planning, okay, in terms of everything what you achieve, but you don't look at the right way. So just this video to say that you'll have to look some right way to approach things. If the things are going to be done in a right way, probably it's not going to be an option at a later point. A quick a scenario, I would put it in terms of a scientific audit, which usually happens. Okay, people, we think that we end up publishing very good impact factor publications. Some novel areas where probably the organism is zoonotically important. We think that the agencies are not looking to us. Okay, I will just put a small story before I start. There was an institute where a researcher was working on a particular virus of human importance. Very good researcher with good background, good amount of funding, approvals in terms of everything which has been happening, with good collaboration abroad. So the people from abroad also came back for a work here. So they have done everything, good publications have come out. 2-3 years down the lane, these materials have to be returned to an agency in terms of a repository, which the investigator also has done it. And then one fine day, the Ministry of Environment and Forests puts a request to DBT, go back and do a scientific audit in terms of the lab. Okay, so we were asked to go without any information, to go back and look into the data there. Just because of one publication which came 2-3 years later, in terms of a collaboration with the same people, where the material already used were already deposited to a national agency and nothing is available in the lab. That was the claim. And now the publication comes in. So we go for a kind of a scientific audit, look at all the requirements there in terms of biosafety requirements, how the materials are handled, when the materials were stocked. There was clear documentation. The laboratory has done clear documentation on some terms of samples, how much were there, how much vials were taken, clear documentation was there. And when it was submitted to the national repository. Now I go back and find after 2-3 years that investigator is not there in that institute. It's a very premier institute. So which means some kind of a small negligence at some point of time. We don't expect scientific audit to happen in this country. It will happen in terms of situations where we try to highlight ourselves in area where of critical importance. So with that caution, do right things in the right way, so to generate the required results. So this is the thing. So before I started, I just inquired with Magesh, what should be the kind of an audience, what kind of a questions you can come across. Okay. So a couple of things which Dr. Magesh sent to me. What type of work requires kind of an approval? What kind of a biosafety practices I need to handle in the lab? So what is the kind of a classification you have? What type of projects requires an approval from the RCGM and other regulatory bodies? Precautions to be taken while handling the risk group 2 pathogens or any pathogens and the genetically modified organisms. What product requires an approval from IBSC or RCGM for an import or export? If I try to import something, if you want to export something, what is the procedure to get an import or an export approval? Formats which need to be submitted to the IBSC or RCGM for R&D, import, export, as well as exchange across laboratories. And what approvals are required for large-scale production of GMOs, especially like a couple of projects which the institution has in terms of transgenic modifications and its field testing. So these are kind of a wide range of questions which has to be taken care if I try to answer in this presentation. So I would like to just put that into 4 major areas, like knowledge on the guidelines. This is very critical. The responsibility lies in this institute with the member secretary first. The member secretary has to be updated in terms of what is actually there in terms of the guidelines. Next comes the DBT nominee, who is going to be in the institute, who is going to be the nodal point in terms of interaction between the institute and the review committee on genetic manipulation. Then comes the internal experts, the external experts, a critical person in terms of a biosafety officer, and finally the chairman. So chairman is not directly actually involved, but chairman takes the responsibility of what happens. But however, the entire driving force in this is the member secretary. Okay, so something in terms of the guidelines. So regulatory resources, where do you access that and what is the availability? Some kind of revisions have come in so that the institute gets more responsibility, because like everything, we need not go to the central nodal agency to get an approval. That can be cleared at the level of the institute. So what kind of revisions are come in so that the procedure is very simplified. It can be done at approval at the institute itself. Some kind of an information alone can be passed on to the nodal agency. And the basic biosafety practices, monitoring and disposal, what actually the IBSC has to look into the laboratory. Okay. So when you look at the biosafety regulatory framework, only there are 3 major things. Probably the Environmental Protection Act is one which you're already linked up to. So the Environmental Protection Act of 1986. So the MOEF rules of 1989. And with respect to recombinant research, the initial guidelines came in 1990, and that was adapted to some extent with modification in terms of plants, transgenic plants in 1998. So these all exam 1, 2, 3 are the major ones in terms of the guidelines that dictate if I'm trying to look at a genetically modified organism or cells, if I'm trying to look at hazardous organisms, usually they say product thereof, anything which is derived from that. If I'm trying to have a genetically modified organism, any product that comes in, any kind of hazardous organism, that product comes in. So the products thereof also coming from those sources also covered under this kind of a guidelines. So when you look at these two, the 3 basic guidelines that dictate everything is included in this regulatory framework. So how do you access? There is one stop portal that is available. So the Biosafety Knowledge Portal, which comes under the Ministry of Science and Technology under PBT. So this is a portal where when you access this, you get all the resources critically required for your biosafety research in terms of rules, in terms of policies, in terms of guidelines. So the department has created it somewhere in 2019. Now they are coming with a revision because there is some kind of a lag in terms of application that has to be submitted here. So probably by early next year, the new format will be released, which will make people to do it effectively in terms of uploading also. So when you look at this portal of IBKP or Indian Biosafety Knowledge Portal, there are two things here. One is from the portal of logging into the portal of an institutional biosafety committee. Second, in terms of what happens when you want to do something to RCGM, and something in terms of databases. So when you look into the left side of it, there are a couple of things which people can get access. So in terms of rules, acts and guidelines, what are the committees and what are the additional formats in terms of submitting some formats to the funding agency or in terms of submitting to the review committee on genetic manipulation. They have given some example formats also. Okay. So this is one portal which everybody should have a kind of a quick access and look at what is there. Second, there is one guideline which dictates in terms of all the regulations pertaining to recombinant research, as well as the containment. Containment, we think that whenever we are handling only hazardous organism, it comes to containment. It also a containment that relates to risk group 1, risk group 2 organisms also. So usually when you look at risk group 2, they cause a disease, but it can be treated. Okay, so when anything beyond risk group 3, you think that containment is required there. As what our director in charge and the IBSC chairman are saying, even for risk group 1 and 2 in terms of disposal, you require specific containment requirements. So this particular document in 2017 lists in terms of the first requirements, what are the classification of the organism in terms of risk group 1, 2, 3, in terms of plants, in terms of animals, in terms of fisheries, everything. So when you want to go back and look at what type of a risk group organism I'm looking at, I'm handling, this gives you the list in terms of the annexure. So you can go back and look at whether my organism comes under a classification of risk group 1, 2, or 3, as per the recent guidelines. It also gives you in terms of risk assessment of the genetically engineered organisms. It also gives you a list of commonly used host vector system that is used for the recombinant research. It also gives you in terms of simplified procedures, how I import, export. If I want to exchange across labs, how do I do it? And also a checklist. Checklist means like usually this comes for a pharma industry where they try to produce a product from a recombinant. You think that most of the product that comes for human health research is natively produced. Almost like 70% in terms of all the antibodies are only by recombinant means. So which means it's a product thereof after the recombinant research, which means there's a huge amount of checklist which the company has to follow through before the product comes to a preclinical toxicity and goes into a human for a phase 1 trials. So there's a huge checklist which the industry has to do. And this is one of the major activity which RCGM does actually. So when you look at the infectious organisms to the IBSC, there is a kind of a revision that has happened in 2021. They have updated because like people working in different area can go back and look at, because like this list, whatever the regulatory agency has put in, is only an exhaustive list which classifies them. Some scenario, as a scientist, you will have a lot of data with you to say that this particular organism is a risk group 3 organism, but can be handled in a biosafety level 2 laboratories. Or biosafety level 2 is nothing but you have a good amount of entry checkpoints, and then you have a classical biosafety cabinet, type A or type B, where you handle the organisms, whatever you require, and then you have a classical disposal that goes in. Okay. So risk group 3 organism, but it is attenuated. You have good amount of scientific evidence for that. And this can be handled in a biosafety level 2 laboratory. If you have a scientific evidence, the organization usually accepts that. So RCGM is a kind of a scientific body where any kind of a document that is put into the committee with scientific evidence, it is appropriately explored. And if the condition requirements for a lower risk group handling, that is also accepted. So the list is not a very defined list. The risk can be flexible with appropriate scientific evidence submitted by the PI. Let me put one example. Vesicular stomatitis virus is a virus which belongs to the rabies virus family, Rhabdoviridae family. Okay, so this virus is used as a vector for delivery of target genes. Example like your adenoviral vector of your Covishield. Vesicular stomatitis virus is also one which can be used for delivery. There are a couple of products which are in the pipeline using this as a delivery vehicle, but it belong to a Rhabdoviridae family. So which means it's neurotropic. But there are specific strains which are not neurotropic, attenuated, which does not cause the classical symptoms. Handling requires a BSL-3, but these attenuated strains can be handled at a BSL-2. So which means the agency puts appropriate information. The scientists who are working in that area, if they have scientific evidence, looking at different research articles, if that is appropriately can be downgraded to a biosafety level, it is possible, agencies accept it. So for the IBSC, the list of the infectious organisms as per the updated list has been revised in 2021. So if you go back and look into the 2017 guidelines, at the last, the annexure gives in. But when you go to the IBKP portal, go back and look into the acts, rules, and documents, you will find the revised list. So that list will be a kind of a guidance which tells that it comes under risk group 1, 2, or 3 in terms of the revised classifications. So the new one which has come in this year, biological research regulatory approval portal. What has happening in industry is when I submit one for a recombinant DNA research, I go to RCGM. And then when I go for a clinical trial, I go to CDSCO. I go to IAAC for getting an approval. I go to Animal Ethics Committee for an approval. I go to the Human Ethics Committee for an approval, which means there are several agencies which play a role in terms of getting approvals throughout the pipeline. So what the ministry thought was, let us put a kind of a single portal of entry. And then when somebody registers, put with a lot of questions on the product development. Based on the questions, it gives you, these are the areas where you get the approvals. So this is usually a kind of a scenario where it is a product based. If some institute or some industry, which is already having an existing IBSC and then wants to go for a product development for field purposes. So probably a registration in this portal is mandatory from this year. Actually, there are 2 OMs in this. So this came with effect in terms of July 7th, 2022. If you go for a product development that requires a C1, I'll come back later what is a C1. So if there is a C1 which requires a kind of an approval in terms of a product development, so this is one portal where you log in, create a registration as you do an IBSC, get the approval so that it gives you all the links. So for this product approval, I require what are the things. Do I require an RCGM approval? Do I require a GAC approval, an NBA approval, CPC approval, IAC, HMC, anything in terms of what are the things that are required for that particular product development? The link comes in terms of the questions what you answer when you register. So this is mandatory from 2022 July 7 in terms of industry or an institute which wants to go for a product development and you go through the C1 application of IBSC. So when you come into the different agencies that are available, okay, so there are 6 major agencies. Just put them as one on the top. You can tell them something as advisory, something as monitoring, something as only regulating. Okay, so when you look at advisory, the central agency, which is going to be the Recombinant DNA Advisory Committee, is an advisory. So it only advises. It gives guidelines, it gives policies. That's it. But the critical thing comes in between the 2nd, 3rd, and 4th. Okay, 2nd, 3rd, and 4th. If you look at RCGM and GAC, so this is created by both the Ministry of Environment and Forests and DBT in terms of the committee. So it plays a role not only in looking at regulations, it is going to be monitoring, regulating, and also doing some kind of a scenario where scientific ethics are required. So this is a central nodal agency in terms of regulatory function. So when a central agency is there should be something which is going to be connecting to the institutes, the Institutional Biosafety Committee, which is in turn created by the Department of Biotechnology, Government of India, looks at a regulatory function, also monitoring and approval. So the RCGM not only regulates and approves, But at the institute level, the institutional biosafety committee is one which looks into the proposal, it goes to the proposal, it monitors the activities, it also approves it. So which means, leave alone the central agencies, the nodal point at the institute is one which has to be clear in terms of how I'm operating it. Because you have a lot of powers now in terms of approval. You need not go to the RCGM for a lot of approvals. Everything has come in terms of approval at the IBSE. You just need to pass on the information in terms of a minutes as an information item to RCGM. Even import and export with limited quantities. Okay. So the committee of IBSE also has the regulatory function. The state level committee and the district level committee usually comes in terms of monitoring when there is a field experiment for a kind of a genetically engineered plants. If you look at transgenic fishes in terms of field applications where field studies are being done, so then these two committees also will be coming in terms of getting your information passed on to them from RCGM, and they might be monitoring you in terms of the activities. So just quickly, what is available in that portal? So the entire stretch of things is there. So you look at any kind of guidelines list. So any person wants any kind of a clarification on classification, risk group, everything is available in this portal in terms of different activities. So in order to support even the COVID research, the RCGM came out with a kind of an empowered committee to fast track. So in the case of pandemic, how the regulatory body acts is, it comes with the kind of a synchronizing the bodies together so that all the proposal goes on a fast track basis. So we were a part of the empowered committee from 2020 April. So any R&D application comes in has to be approved in terms of starting the work within 48 hours. So any laboratory which is starting a work in terms of recombinant research pertaining to COVID starts quickly getting the NOC to start the work within 48 hours. So they have an option to come back with all the data as early as possible in terms of an application where they put all the data. So all the things which was possible now in the country in terms of the pandemic within a 1 year as a kind of a scenario, not in terms of the regular conventional vaccines, I'm telling recombinant vaccines. Some conventional vaccines where conventional products research requires high safety level, example, the COVID, where you require a BSL-3 facility to handle. So only that part of kind of a clearance in terms of the handling of the virus comes to IBSE and the RCGM for information, but other activities goes to the different agency. But in terms of all the recombinant work that has gone on, in terms of anything available in the market, in terms of a current vaccine, which is approved, emergency approved, all has come out in 2 years. So which means the regulatory agency also looks at a pandemic scenario to fast track things. So it's not a kind of a set deadline for every activity. Okay. So people have to be aware of all these documents, not completely, at least meet the member secretary, get things done. Okay. So member secretary has great responsibility in terms of getting things updated. Okay. And why the IBSE is very much important is there is a guideline which came in 2015. Okay, revised simplified guidelines. So which has been revised in 2020, which means earlier when I want to send something outside in terms of a recombinant molecule, I want to send a plasmid outside. I want to bring a plasmid from a different lab here. I want to send a kind of a microorganism which has been grown in bulk, inactivated or alive to a different laboratory. I want to get a bulk of infectious virus from different laboratory. Not a kind of a regular thing, but in bulk in terms of infectious organism. So the IBSE is empowered with the limitations here, which means you put the proposal to the institutional biosafety committee, and this revised simplified guidelines comes as an annexure in terms of plasmid, how much quantity you can approve. In terms of proteins, how much you can approve. So you need not wait until the application is submitted to the review committee of genetic manipulation, which is the central nodal agency. You get the thing approved, get the permission, get the NOC, that is not required. So for an institution, please keep that in mind. As per 2020, in terms of import, export, exchange across laboratories, this simplified guidelines tells you with a limited quantity, you can approve. Once you approve, there are a couple of things which need to be done. So you will have to look at, okay, I have approved it. I need to put the minutes, put it to RCGM for information. Second, every year in terms of an exchange. Probably usually what we do in our university is any plasmid we get from other laboratory, we try to document that by the end of the year, there's an annual transaction record. So this annual transaction record, if it's going to be submitted, it makes things easy. That's it. Which means where an investigator puts a proposal, puts it to the IBSC, IBSC looks into it, approves it. You need not wait for the time for the clearance from the RCGM. Okay. Second, in addition to the recombinant thing, organism which also comes under comet category. So anything which is going to be safe in terms of handling, that also can be approved at the level of IBSC itself. Post import or export, you just need to update, which means the institutional committee has more role in terms of maintaining things here. That is why probably the IBSC chairman is more worried on to what is happening in the institute, because more responsibility lies with the institutional biosafety committee now, rather than getting the information done to the central nodal agency for approval. Otherwise, I prepare the thing and then put it to the central nodal agency and say, let us wait for the permission. Now, the institution itself clears it. If the institution clears it, the institution would like to see that things are happening in an appropriate way. So probably the chairman's worry in terms of small things also is because of this. Okay. So the IBSC has more powers now in terms of operating, in terms of approval also. So just coming to the point of the institute. Okay, let me just put some figures out here. Probably the scientists can keep quiet. The people at the back can tell any problem in this photograph. Probably our laboratories are very good that we have wide amount of space. People have an entire workbench to work, but this is not usually happening in Western laboratories. Probably the research fellows should say, what is the, any problem is there? Everything is fine in that photograph. You'll have to come out with an answer. There's no workplace. That is the only problem you find. You cannot touch anything here. The classical thing which will happen is your experiments are not going to be appropriate at all. You'll end up with a lot of cross-contamination. You do a PCR, your NTC will always be amplifying. Okay. Second, this looks very neat. Any problem here? This is a classical biosafety level 2 cabinet, biosafety 2 cabinet with good things arranged inside, appropriately given. Any problems? It is very neat. Pipettes are outside, but that doesn't matter. It is only in the area where it's having that air layer. When you look at the biosafety level 2 cabinet, it's a kind of a scenario where like you enter a shop where there's an air shower. That air shower is going to be a kind of a scenario where it filters. That air shower goes to the pocket that is going to be down, circulates and comes as a kind of a filtered air. So it's not a problem there. Anything else? There is no place to work at all. You have kept things appropriately, but if you want to handle things, you cannot handle at all because like everything is popped up inside. There are pipettes which can be stored outside because it comes with a kind of a sterile cover. So you need to have appropriate space in terms of where you're operating. Any problem here? Do you have this cabinet in your lab? What is this? Fume hood. So any problem here? That is one. Anything else? I've not put that image actually. Actually, when you look at all the liquids in terms of organic solvents, they have to put in a secondary containment. So you have to have a kind of a vessel or a tray where these are separately sorted out. So if you're not doing it, anything breaks, there is going to be leakage that is going to be corroding the surface area. So if you're having a kind of a containment in terms of a tray, which is going to be effectively stored there, liquids has to be appropriately assorted and then stored in trays. So these are things which we regularly don't follow at all. This probably one of the things which happened in our earlier IBSE chairman was there should be an undertaking from every lab in terms of spills, in terms of risk assessment, in terms of accidents, in terms of regular handling. There should be some display in the lab, and they should give an undertaking that I follow it. I'm regularly following it. And one of the thing which we practiced in our lab is the chairman was telling about the gloves. The director also was telling about the gloves. Within the laboratory, across the compartments, because like at some point of time, you may not be using the gloves. So we have two things in the lab. So the top will be a red spot where with glove. So there will be always a sticker on the top, red, and bottom green. So with red, with a glove, you use that area to push or to pull. The green one without the gloves. So if people follow this, this has been classically practiced even in HIV labs. You go to a lab where critical things are done, they will have a red area where they handle with gloves. With the gloves, they handle only in the green area. So this is also one. And in our laboratory, for all the gel handling where you use ethylene bromide, the person has only the left hand gloves. So this only goes into the tank, the pipette, the keypad, the documentation, everything happens with the right. So that this ethylene bromide doesn't go into my right hand. So small things in terms of application, if people refine, people also in our lab, always they post, put your boards appropriately in the place, put your combs appropriately in this place. You remove the gel from the sink, discard the gel here. So all these things depends not only on to the researcher, but also the people who are going to be in the lab in terms of seeing that my workplace has to be effective. My workplace has to be neat. In our laboratory, there are two people who always do that. Research fellows, even though young, good, they try to put writings, good writing, so that everybody does that. Everybody follows that. Otherwise, they come and complain. Sir, I'm facing the problem. So please make your work culture in such a way that it is not problematic to others. It is also effective. And somebody comes into the lab, they clearly see that and say, I will have to go and do that in my lab also. I have to implement it. So that should be a scenario where the research fellows have to take a responsibility in terms of implementing things in the lab. Things are available. Implementation depends only on the research fellows because young minds are very active. Okay. So let's go to the topic. So when you look at laboratory safety, put the radiation aside because like none of us are working with radiations. So you require laboratory design in terms of how you enter, what are the things there. You require personal safety, what are the materials you're going to use. In terms of biological safety, in terms of chemical safety, finally the disposal safety. So a laboratory has to look at all these things in terms of safety. Okay. So when it looks into how the safety procedure is implemented, so the kind of a book that or the guideline document that drives all these things is the Institutional Biosafety Committee Handbook. So this was earlier revised in 2011. Now 2020, the revised edition has come in, which has given all the revisions in terms of what the Institutional Biosafety Committee can do it. So the member secretary should have a copy of this document always to go back and refer. Okay, so this handbook gives entire thing. So I need not go back and look at things again into the net. So what is the Institutional Biosafety Committee? So Institutional Biosafety Committee is not a committee alone. It's a statutory committee, which means it is approved by the Department of Biotechnology through a process where they are looked at the institution's capability, institutions things in terms of research, institution things in terms of containment, institution things in terms of disposal. So a statutory committee means it has more powers. It has been defined with the powers by the central nodal agency to look at on its behalf to operate at the institute. So when I say a statutory committee, so you have more powers in terms of saying that this has to be done like this. I want this to be done in this institute following these protocols. There can be strict norms implemented by the IBSE in an institute to go back and look at things even regular, non-hazardous to humans have to be handled only in this way. So it's a kind of a statutory committee, not only into recombinant activity, I'm just putting it into line, because in our institute, if you look at all hazardous organism institute in terms of risk group 2 and risk group 3, risk group 3 organisms which can be handled in a biosafety level 2 also, Example, I handle with a kind of an isolation of salmonella. Look at all the antibiotic resistance. It comes to the IBSC committee in our institute. So not alone the recombinant activities, anything in terms of hazardous microorganisms also comes under the institute in terms of how I want to monitor that. Okay. And if you're going to look at, it's a nodal point, it implements, it conducts on-site evaluation, it assesses and monitors. So that is the power of the committee. We feel that it is not a policing job. It tries to implement good ethics in the institute for the people to follow. So there has been a kind of a revision in this. When you look into the composition, I've just highlighted the people in the middle. Okay, the middle group are always the group of interesting, has more activities to play in. The chairman sits on the top, tries to look into in terms of the institutional capability. He comes in whenever there's an issue in terms of how do we operate. Otherwise, the operation depends upon the three people who are sitting in the middle. The critical driving force is the member secretary. The critical driving force is the medical officer, qualification medical person who is going to be a biosafety officer, and the DBT nominee. I would say the member secretary and DBT nominee should have a good amount of sync in terms of the guidelines. Sometimes the member secretary may not know it. The DBT nominee will support them. So anything which is going to be a situation of audit, usually they contact the DBT nominee and then they come to the institute. So the choice of the DBT nominee and this action is one critical event in terms of entire thing. Okay. Members, critically, if you have good members, they can support you in terms of activities. So it's for 3 years and you will have to renew it every year. Now it has been mandated, which usually most of the IBSCs don't do it. Every year you are supposed to give the annual compliance. So this year, how much projects have gone in, how the activities have happened in the institute, any spills, any containment risks. So that annual compliance has to be given. And the medical assessment also has to be given because some laboratories will be having hazardous organisms of human importance. So medical complaints and annual complaints has to be uploaded in the IBKP portal. Otherwise, any proposals which requires approval from the central nodal agency will not be cleared at all. So annual complaints, the IBSC can make it mandatory that you submit it in the month of January. So that is going to be happening for that previous year. So January for 2023, if you're trying to do it in 2022, that completes the entire process. Any application which goes for an approval for the central nodal agency is very clear because they require the annual complaints. The annual complaints of institutes, every meeting of RCGM, the institutes are displayed. So these are the institute who are given the annual complaints, the medical assessment also. So kindly submit the annual complaints and the medical complaints in the month of January so that any process that can be supported will be easily done without any kind of an intervention. So what the IBSC has to do? Okay, so it looks at three major things. One is to categorize the entire research into three major aspects, category 1, 2, and 3. It categorizes them in terms of risk group, in terms of the containment requirements. So these are the three aspects which the IBSC committee has to look into. So category one, if I just wanted to put in, usually this does not require an IBSC approval at all. But when you go back and look into the document, it says, I don't require an IBSC approval. But however, the institute can put an question onto that and say, I just want to monitor that. Please put an application alone so that I know this kind of research is happening. So I isolate a bacteria from a source, which is going to be interesting, which is going to be having virulence in terms of some effect in the organism which you are studying. So that is also a scenario where it is not potentially hazardous to human beings, but it's potentially infectious to the animals which you are handling. Probably escape might have kind of an end. So in that scenario, so that has to be categorized as category 1. If I'm trying to isolate, I'm trying to look at antibiotic resistance, I'm trying to amplify that in large volumes, it comes as a category 1. So the researchers who are going to be in terms of research fellows who are going to support the investigators have to put the application in terms of categorizing them, which will be easy for the IBSC also. So you categorize the proposal as category 1 if I'm looking at a self-cloning experiment where I'm trying to amplify an isolated organism. I'm trying to have multiple organisms as a mixture, which is going to be having one virulent organism also. So category 1 is nothing but a self-cloning. Category 2, which comes under 2, 3, and 4 risk groups, as well as self-cloning. You are trying to put in some target gene into a bacteria and try to amplify that alone. So anything between category 1 and category 2, the IBSC can approve itself. You need not go to the nodal agency to approve it. So if the investigator puts, or if the research fellow says that my application is category 1, just go into the application to look at what is required, clear them from the institute itself. Don't send it to RCGM if it's going to be category 1 and category 2. Even category 3, IBSC can approve in terms of case-to-case basis. So if it's going to be a category 3 experiment, we'll come to the applications later. Category 3 experiments have to be submitted to the RCGM for an approval. You get an NOC and then only you start. So now process has been made very simple. Once you do the minutes, you approve the minutes, upload the minutes into the IBKP portal. Once you upload that, along with that, you submit the form C1. So for the member secretary, you log into the IBSC portal, request for RCGM access. Otherwise, the RCGM access doesn't get deactivated. So you log into the IBKP portal, request for RCGM access. Then in your IBSC portal, sir, there will be 2 tabs, one IBSC, one RCGM. So if you submit the minutes, click the RCGM tab in your IBKP portal, submit the form C1. So what happens is when you put the form C1 with all the details, this is taken up by the central nodal agency. It goes to the application in terms of all the attachments. And if it's going to be appropriate, it says, okay, then you upload the signed copy. So if you have any kind of a problem in this, so I'm there to support you. Okay, so this is a scenario where it happens automatically, but the central nodal agency might take a week or 10 days because there are almost 900 IBSCs throughout the country. So 900 IBSCs on a day-to-day basis in terms of approvals takes time. So probably one which we can support the institute is the DBT nominee can directly support in contacting the central nodal agency to update on to what we have uploaded and then get that approved. Otherwise, we can support that in from our end to go back and say that things have been done. You have uploaded the form C1. Kindly look into it. If it's going to be okay, they get back in terms of a notification. Now you sign the upload, signed the copy upload. Yeah, no problem.
Many times when you see like routine work or bacterial isolation is also routine work. So in that case, every year one has to take the permission.
Actually, what we have been doing in our institute and what we suggest also is a scenario where it's a continuous process of some kind of a bacteria trying to isolate. Okay, and try to look at the monitoring, look at some kind of an antibiotic resistance profile across the time. You submit one application at one point of time, get it approved in the IBSC. By the end of the year, the next IBSC comes in. If it's going to be a 1 year period, you just inform the IBSC alone that the project is continuing. So that the IBSC has a kind of a thing saying that any others. So this project is continuing in terms of the same criteria, what are being there. The presentation is not required at all. Every time you need not put the application. So this can come into the category in terms of any others and then just update in terms of the activity that is going on. Otherwise, people request every time application, every year application presentation. This is going to just waste time. So things in terms of updation can be added in any other items and then say that the project approved is continuing. That's it. Just to have an idea in terms of the central nodal agency, if something is happening, they want to go back and look into it. The project is continuing. Why? Because some of the funding agencies insist on IBSC approval for your proposal. So at the point of time of completion, if they are looking at the approvals, if it is not there, then that might be a kind of a query. So you just put that and ask the IBSC alone to put that into any others. Does that answer, sir? So categorizing the application has to be one which the IBSC has to do. Second one, it looks into the risk group. So when it looks into the risk group, it looks into scientific considerations. It is really a risk group 3 pathogen, but I can do that in a class 2. If I say, example, rabies is a risk group 3 pathogen. I don't require a BSL-3 facility to handle it. I can do that with the BSL-2 cabinet itself. So which means, so there is a scientific consideration that has to be put by the committee to go back and say that this can be done in a biosafety level 2 itself. So It is not a general consideration that has to be taken into consideration. It is a scientific consideration that has to be taken to look at risk group classification also. So this has to be done effectively by the committee, looking into the containment facilities, what you have, what is the ethical issue there. Last but not the least, this is a scenario where the pyramid, actually risk group 1, biosafety level 1, category 1. So anything until category 2 with risk group 2 requires a regular lab with a biosafety level 2 cabinet that is more than sufficient. So anything beyond that requires a biosafety level 3. If you want kind of a biosafety level 3 work to be done, so our institute has a BSL3 level laboratory, which you can approach for any kind of an experiment. You can also approach them in terms of small activities where you want to challenge. Probably we don't have a kind of a challenge facility there. We have a challenge facility in terms of animals, like a small animal like mice, rat, rabbits, chicken. But in terms of fishes, it has to be modified. But however, if you want a BSL3 experiments in terms of in vitro purposes, you can approach our institute in terms of the facility. Okay. But otherwise, all the regulatory agency looks at anything which is available in the country, whether it is also available for other agencies also. It is mandatory from last year that all the biosafety level 3 laboratories has to be certified by the nodal agency. So any laboratory which is not certified, there might be a kind of a scientific audit to go back and look at why this has not been done. So risk assessment is one which we usually don't do anything in the application. When you go back and look into the application that is put for the IBSE, the risk assessment is one which people have to fill in appropriately so that the people in the IBSE or the committee, IBSE committee can go back and classify them appropriately. So risk assessment is one. And for the people at the back, always look at the primary barriers. Because when you are handling, what is the barrier that is going to be preventing you in terms of operations? When you are handling some kind of an infectious agent, even though it is not hazardous to me, what is the primary barrier which is going to be helping you in terms of personnel, in terms of product, or in terms of environment? So this is one with the IBSE also has to go back and monitor. If my containment, primary containment that is going to be available in the lab in terms of primary barriers, is it appropriately evaluated in terms of its validation? Because a biosafety cabinet, if not appropriately evaluated, it can result in contamination. It also can result in kind of an infection. So whether it's a regular laminar flow or a class 1 or a class 2 or a class 3. So primary barriers is one. So the research fellows have to be looking at how do I operate in a kind of a primary barrier. Usually people in our lab put, these are the authorized people. And when you operate here, there will be a kind of a material which is kept inside to dispose. And what you do after you finish your work. So kind of a written work onto the primary barriers will always help the people to work there effectively in such a way that whatever comes out is effectively decontaminated and disposed. So primary barriers is one which the committee also has to look into. It also has to look into all the safe work practices in terms of gloves. We always, when we handle highly pathogenic organism, we always use a double gloves. So that when in the laminar flow, we use double glove. When we come out, we remove the first glove so that your hands are fine. Now we go into the laboratory, not outside. We do other things in the laboratory, touching all the centrifuge and other things. Come back, put the new glove, operate. When you take outside, you remove the first glove. So that the other instruments in the laboratory is not contaminated. Your operation within the biosafety cabinet alone has the infectious potential. Okay, so in terms of disposal, in terms of liquids which is coming out, in terms of liquids, when you're trying to suck that through vacuum, you have a kind of a filter attached to it. So all these things are routine things which the laboratory also has to be taking care, and which the IBSE also monitors. In our laboratory, we put all these things, written guidelines. Second, in terms of segregation, so what kind of a segregation you need to do? Mostly I feel that your things will not come into a category of red. So it should come into a category of yellow, which requires a biosafety disposal in terms of a third-party carrier. Otherwise, mostly it's going to be a scenario which is going to be safe in terms of blue bags, where you just autoclave and dispose it. Otherwise, disposal through a third party usually comes for an yellow ones. Segregate that and keep it. And this is a regular one which we use in our lab. So spills, we use 10% bleach. and general disinfection 1% bleach. So this should be effective in taking care of most of the infectious agents, even it is not going to be infectious to us. Best options to even something spills down, put 10% bleach. In the operation area, 1% bleach. Wherever you find something to be disposed in terms of liquid, 1% bleach inside the laminar flow in a bottle can help you in at the point of decontaminating whatever you're handling. So good option. What we usually do is all the media bottles and the serum bottles what we come in. So it's a kind of an autoclave bottle. So we collect that. One bottle will be always there in the laminar hood with a 1% bleach. So you directly discard all the things, not plastic. Plastics, usually what happens is if you put it in the bleach, people will go and then throw it in the sink and that clogs. So plastics are collected separately, autoclaved and disposed. Any liquid goes into the liquid, not into the pipettes after usage doesn't go into the beach. So usually that will be very difficult for the people who is going to be attending the lab in terms of cleaning, will find it difficult to decontaminate that and dispose it. So even they are going to autoclave it, they'll have to collect the plastic separately. So you have a separate autoclavable bag inside where you put the plastic, and you have a bottle where the liquid can be disposed. So at the point of decontamination, the cover alone gets tied up and then goes for a decontamination by autoclave. So look at all the critical things required. Don't think that this organism is not infectious to me. There can be potential things which can happen, which we are not studied at all. We think that these agents don't potentially cause an infection, but probably over a period of time, some kind of a transformation that happens, bacteria can change because they can accumulate a lot of things in terms of plasmids which can help in getting antibiotic resistance. You will have things here. So don't think that I don't require the basic safety. Basic safety because you are here for 8 hours. The time what you spend in your home is less. So 8 hours in the lab has to be a place where it is going to be safe, happy. So basic things in terms of a gloves or a kind of a goggles or a lab coat always supports you. So some spill, you are going to corrode yourself. You think that my handling is very strict. I don't think that it's going to fall onto my shirts. It will happen at some point of time. So take care of all the general laboratory things in terms of gloves. Specific requirements are there for specific kind of corrosive materials. So ask the laboratory to get that materials. Example, if I'm trying to use an organic solvent, I use this glove type. So usually we use a nitrile gloves, which is going to be for the routine purposes. In terms of other things, you need to have some kind of a special gloves that is always near your fume hoods. So there are also 4 things. Whenever you're disposing, disposal material always should be closed. They should be appropriately labeled so that the person who is going to be attending the lab in terms of cleaning, even if he's not going to be aware, that label tells him that I need to go take it to an autoclaving position. That label tells that I can go back and dispose it safely. Okay. So in terms of the things what you label, lid or leaks or location should be appropriately labeled in the lab. So all these things are done. So now let me just come to how the regulatory agency operates in terms of approvals. Assume a scenario where one of the person is putting an application to do a kind of a recombinant protein expression in the lab. Okay. So I don't have option to go back and amplify that gene. I am going to go back and synthesize that. Okay. Synthetic construct where I give it to a company and then get that synthetically synthesized, put in my vector of choice, and I'm going to get it here. Okay. You think that you give it to a company who's a distributor here, but this person will give it to a laboratory outside. Usually gene synthesis happens outside. Critical thing which we came across recently. There is a virus which affects cattle. Okay. It does not affect the humans. It's a kind of a pox family virus. We thought that, okay, we get the IBSC approval and give it for synthesis. The agency comes and says that it's a potential threat in terms of synthesizing that particular gene. So none of them synthesized for us. Why? Because that is the kind of a virulent gene of a pox family, which can potentially be used for other sources, all other biological activities. So we didn't think that it's a kind of a pathogen of only livestock importance. It is not going to be harmful, but it can have a kind of a potential thing in terms of biological threat. So when you put a kind of a plasmid gene for synthesis, you get a kind of a IBSC approval that I'm going to do this experiment. I'm going to clone this gene. So for this gene, I'm just going to get a puck vector with the thing synthesized. I transfer it to the expression vector now. Okay, so this is going to be imported. So the best option, what you do is you put the application to the IBSC. Let the IBSC get the form B1. Form B1 is only for import. So when you go back to that IBKP portal, into that, in terms of application formats, when you click that, all B forms are related to us. So B1 is for import. B2 is going to be your NOC, what comes in. B3 is for export. If I want to send some plasmid from my laboratory to some other laboratory outside the country. So I can, sometimes I may not have money to synthesize. I will get a plasmid as a gift from some other laboratory. Okay. So get that form B1 submitted to IBSC, get that approved by the IBSC itself if it's going to be 1 milligram plasmid. So until 1 milligram, the IBSC itself can approve. Get that approved by the IBSC itself. If you want to send the plasmid to a different laboratory, the same thing in a form B3. So all in that revised simplified guidelines in terms of how much is permitted. Okay. So if I am trying to get something from the repository, example, there were a lot of laboratories which were working on COVID. So they have to get the kind of strains from a different laboratory. So getting strains from across different laboratories, you put a form B5. If you want to transfer it to a different laboratory, put B7. So import, export, getting and exchanging. So all these things can be approved by IBSC itself within that permitted limit. Okay, so which means when along with the application, put that proforma, get that proforma also. So like what IBSC has to do is when the application comes in, you just go through the application. If something is there in terms of the application formats, get that application format also approved along with that so that you approve it.
So this one NBA doesn't play any role.
In terms of all the import, NBA comes in when you want to get a kind of an organism. This is only in terms of approval pertaining to RCGM. Okay. So there are other bodies where I put it into the first slide where that portal, single window portal is there. If it requires some microorganism to be sent outside, a whole bacteria to be sent outside, you require the NBA approval, NOC from the NBA. So this is only the approval pertaining to the biosafety practices. Any other approvals required from other agencies are also required. Example, I'm getting a virus, hazardous virus from BA Resources. BA Resources is an agency from US which collects things. I can put an application and get it. So which means, so some hazardous organism, I put an application to the IBSC. If it's going to be a highly risk organism, it puts an application. It asks me to put an application to RCGM. So this form B1 will go to RCGM. RCGM will say you are approved with an NOC. But however, to bring that into the country, you require the drug control approval. You require the Ministry of Environment and Forest approval also. So you also have to get the approvals. RCGM doesn't support in that. So any other approvals, I'll tell one scenario which I came across in IIT as a DBT nominee. So they are trying to get a fixed brain of one particular snake, because that laboratory is looking at complete three-dimensional modeling of the brain, looking at the neuronal circuits. So they are looking at how different brain things function. So they are trying to capture images from a fixed brain. So they are trying to get only a fixed brain from a type of snake from South Africa. It's a fixed brain. It's non-infectious. The person says that the person comes here for the conference. Can you bring it? So what do? They went for NBA clearance. They went for MOEF clearance, and then they only approved actually. But it was put into the RCGM IBSC, but IBSC does not have any role at all because it's only a IBSC also has to keep in mind that only in terms of biosafety risk, something which is going to be like an import of material, it need not, I say it's not under my purview. We said that this thing is not under our purview. So you will have to get appropriate approval from other agencies. You need approvals from other agencies if you're trying to export and import depending upon the material which you're handling. This is only a kind of a critical thing in terms of biosafety approvals. So in terms of the applications in terms of form B1, when you need a form C1, I said in the single window portal, all things which you are doing in terms of, I think like there were a couple of proposals here in terms of a transgenic modifications. I have a bacteria where I want to express some thing and then use it as a kind of a feed or something else. So a genetically modified organism under a lab scale. So it's only a laboratory adaptation. So where you put it in the IBSC, get the approval from IBSC, put a form C1 for information to RCGM because you are doing only the lab trials. You're not going to take anything in terms of things to the field. So it's going to be a laboratory evaluation in containment conditions where I'm just going to generate a live modified organism, but not of a potential threat organism. So it's going to be a very safe grass type of organism, generally regarded as safe. If it's going to be a grass type of organism where you're trying to do some modifications and you're doing laboratory scale activities, you clear that under IBSC, put a form C1 to RCGM only for information. So you don't require an NOC. But if you are looking at field applications, any kind of even generally regarded as safe organisms, genetically modified, if you're trying to do field trials, so you put a form C1 initially itself to get the research and development approved, which means my proposal requires an approval from RCGM before I start the experiment. So form C1 is for getting an approval in terms of research and development of a project, which requires a kind of a approval from the RCGM. So you get that approved, do all the activities, come back, and then if you are trying to take a thing to the product development pathway. Okay. So once you develop, you have to put the form C3. Form C3 is nothing but what I have done in terms of the R&D, which I have committed. This form 3 goes back to RCGM and then gives an NOC. Now you tie up with an industry for a product development in that. But anything in terms of generally regarded safe organism, which is going to be genetically modified as a transgenic, in terms of laboratory scale, it should be fine in terms of clearing with an information. But however, if you plan anything in terms of future, even I do now and say that I'm going for a future application of transgenic into the field, you cannot do that. So you pre-plan earlier itself. Pre-plan in such a way that the product development, what I have, has a kind of a potential applicator in later stage. You do that as a form C1 in terms of NOC earlier itself. So that would be a better way rather than going back and then putting a separate application again. But you can do that. The agency looks at already the data, whatever has been generated in the lab in terms of all safety before it clears the C1 application. So now if I go back and put the questions again, can you answer all these questions? What types of import can be done? What type of export can be done? What type of exchange can be done? In terms of exchange, in terms of GMOs, transgenic fish or transgenic bacteria, there are a lot of laboratories which are doing it. There are a lot of laboratories which are working on transgenic plants. They are doing confined field trials because there is a monitoring agency which goes and does that also. So don't think that transgenic work is a kind of a difficult work in terms of regulatory approvals. Regulatory pathways very classically streamlined. RCGM does the activity every 2 weeks. Okay, so I attend a meeting every 2 weeks, not just to attend a meeting. There will be at least 600, 700 pages of a document of a company which is going to be confidential, which comes to a member. So a company or an institute which goes into a product development pathway should not be hindered in terms of regulatory process. So if some query comes in now, you have 2 weeks to go back and answer that query in terms of the response. If you already have supportive data with you, submit that, it comes to the next RCGM meeting. So 2 weeks is a shorter period where the regulatory agencies working with people not full-time. Let me put it, all the people who are a part of the review committee of genetic manipulation are only part-time people. Second, this committee is supported by a biosafety unit, which is going to be a separate unit, which does preliminary screening. So preliminary screens application and put it to the experts. So we will have to go back and look at all the application that comes in terms of why I'm telling form C1 don't put a lot of things there is it automatically comes to us and puts us in a lot of pressure. So that is why the agency said give all the powers to the IBSC, let them take care. If they end up in error, the DBT nominee can help them in correcting it. So they're not going to go back and look at policing them. So DBT nominee helps in correcting that. DBT nominee helps in contacting with a member and gets it sorted out. So things are very fine in terms of streamlined. If you submit an application now to RCGM in terms of clearance, It's a well-defined process. If you look at other things, I'm not sure, but this is very well streamlined. So the only thing which I can support from my end is anything is required in terms of clarification or some kind of a critical thing which need to be considered. I can support the institute in terms of an external member. Putting a small thing about our laboratory. So it's a kind of a funded project which was happening from 2012. Looking at taking some industry experts as well as academia and trying to take small leads and then see that whether we can come out with products. So this has been now in the third phase. We also do some service. Why I just put this is we have a kind of a good in vivo imager with us. So which means some fishes are not actually fluorescent. So if you look at some kind of a bacterial trials or a drug trials where you are fluorescently labeling it, you can use the in vivo imaging system effectively, where you're trying to monitor the thing. So in vivo imager is available. If you want to do a good confocal imaging, so we are open. So facility is available where we invite you to use the facility. So we have been doing with good amount of achievements since phase 1. So we have a lot of technology transfers. At least one has entered into the market. Getting a regulatory approval in terms of recombinant products to the market was a long process earlier. Now we find it very easy because things are happening in a regulated way. So with this, I stop, and any clarifications required, I'll take up questions. There should be a lot of questions from the youngsters actually.
Whether to import grass generally regarded safe organisms, whether approval is required from IBSE?
DRS is a safe organism. Why do you require IBSE role? If I am an IBSE committee member, I'll say it is not under our purview. If you do a modification, is required.
Sir, for human diagnostic purpose, we want to send some samples to abroad countries. In that case, what is the rule and regulations?
So you want to send the samples for?
In India, it is not available for diagnostics.
So one thing critically is if I am looking at a diagnostic approach, human ethical approval is not required. Okay, example, I'm looking at my institute where I do a human testing only for diagnostic purpose. I don't retain the sample for R&D. So ethical approval is not required. So if the sample is going to be for any genetic testing, assume that is going to be for genetic testing, it is not required. You can tie up with a agency which does the testing here. Probably if the facility is not available here, we can do it outside. But in terms of genetic testing or things, a lot of things are available which people don't know. If you are looking at genetic testing for human purposes, n number of labs are there in terms of genetic testing. So you need not send the sample outside. If it's going to be some kind of a bacterial pathogens or something else which expresses something to be sent outside for diagnostic purposes, it requires an IBSE approval, an RCGM approval, and a clearance from the Ministry of Environment and Forests as per the National Biodiversity Authority. But human samples for diagnostic testing doesn't require an approval in terms of sending abroad, but a lot of facilities are available within the country at a cheaper cost. Hyderabad and Bangalore have n number of companies working on it. One simple thing which we came across last week. If I have a scenario where I look at an SNP in a gene, okay. So my laboratory does a lot of things in terms of whole genome sequencing and identifies several SNPs. I want to go back and screen the population in terms of SNPs. So there are a couple of labs where you need not go back and do a microarray and do it. So there's a small system where I put samples onto my left, I put all my primers onto my left side and right side samples. At a single time, 96 samples can be screened for 96 SNPs with a much lower cost. Systems are available within the country. One is available within Chennai, which charges huge, at least in Hyderabad and Bangalore. People who are looking at SNP screening in the population in terms of looking at screening wide number of animals, Hyderabad and Bangalore has options where you pay for it at a lower cost. The cost of screening comes to less than one regular PCR.
Thank you very much, sir, for your great presentation, sir. Sir, I have a few doubts. One is, sir, like we are working on these three pathogens. So most of them are not human pathogens. But still, do you think that in our institute, at least DSL2 facility is, from the scientific point of view, which is necessary?
I would foresee my question or your question in two things. One is the institute not looks at the pathogens in terms of potential risk to humans. The institute looks at potential risk in terms of environment. The biosafety requirement takes care of personal, laboratory, environment too. So in terms of potential environmental safety, it is a concern. So if that is going to be a concern, IBSC has to take up a role. In terms of two things. One is to look at how you're handling it, how you are just trying to dispose it. That's it.
Okay, sir. Sir, another doubt I have that suppose generally regarded as a safe organism for the humans, but may not be safe for the animals, fish or shrimp. If that is the case, and we have two scenarios, to get a pathogen or to get a grass organism from outside another lab, and another one is to import it, what protocol we need to follow? In these two situations?
Yeah. So it's going to be a kind of a it will come under the category of comet. Any mechanical equipment, technologies, medicines, organisms. So comet, if it will come under this comet category, if you're trying to import something which is also considered to be safe, but in one context, but it's going to be hazardous in other contexts. So if you critically see that it may not come under the purview of IBSC, but IBSC can take up the responsibility in terms of saying this is the application. RCGM also will not come in because like this is only an organism, it's not a genetically modified organism. So you will have to get a kind of an NBA approval or NOC to import the organism. So that is critical. NOC is required. And to import, you require two things. One from a drug controller and one from the Ministry of Environment and Forest. So which will be a scenario where you will have to look at a third party to support you in terms of getting the clearance. NBA, things are going out easy now to get an NOC. But however, in terms of getting an approval from Ministry of Environment and Forest, as well as because like the product what you are getting now into the country might become a product at a later point of time. So in terms of a drug control approval also is kind of a required if something comes under that category.
Okay, thank you, sir.
Sir, I have a small query.
Yes, sir.
Normally, when we talk about this biosafety and all, we talk about more chemicals and all these kind of things. Do the liquid nitrogen handling will come under purview of biosafety? Well, this has got a serious hazards actually, if not handled properly, including respirational and the others.
Yes, sir. Sir, I will see that under the category of chemical safety. So in terms of a safety where it is pertaining to personal safety and chemical safety is a part of the biosafety protocol as such, but probably I will not directly put it into the IBSC to look into it. This is a general laboratory procedure in terms of a chemical safety.
General laboratory.
So it will come under kind of a good laboratory practices, and so that is a kind of a mandatory requirement us in terms of all the liquid or organic solvents or other handlings.
Okay, thank you.
Even the biosafety guidelines doesn't mention about this chemical.
So one of the thing is like central nodal agencies cannot take a lot of things into bracket and go back and look at policing it. So it always tries to look at something to be done at the institute. Even the institute level agency also, you are a scientist who is working in some area, but trying to accommodate something in your career as a kind of a support to the institute. So when things are being defined, only critical requirements ought to be put to a committee. Other things in terms of all the basic safety is a procedure which the laboratory should implement. The PI should implement, the PI and the research fellows should implement. They should bring down to the new generation that is going to be coming in. So it's a general laboratory protocol where guidelines cannot come and dictate that general laboratory protocols has to be done like this. General laboratory protocols has to be one which has to be appropriately implemented in the laboratory. Then it comes to the institute. So institute cannot take a responsibility in terms of some agency to come and dictate the guidelines on that.
Dr. Raja? you want
Yes,
to
sir.
ask?
Yes, sir. Sir, myself, Raja Swaminathan.
Yeah.
I am from NBFGR, different institute.
Yes, sir. Yes.
Sir, we are having a center at Kochi. Our institute is at Lucknow. So
Okay.
at Lucknow headquarters, we had a, IBSC committee.
Okay. Sir.
But that when we approach that to the center, also some project has to be approved.
Okay.
DBT told that you should have a separate IBC for the center also. So now we have got the user ID and everything we got.
Okay.
When we are about to submit the biodata of all the committee members in that website, so
Okay.
we could not do it. Reason is our director has got recently changed. So the chairman name, our old director name was there, old director phone number was there, old director email ID was there. When we are conducting DVT to change the chairman, so we cannot get the proper response from them. So can you help to get the change the
Sir,
chairman's name?
I will correct it a little bit here. Okay. So when the IBSC is registered in the IBKP portal, it gets registered in the name of the institute with the particular person existing at that point of time.
Yes,
Okay.
sir. Yes, sir.
But when you go back and look into all the modalities of operation, you can go back and update in terms of the current chairman and the current person who is taking care. So the change will not be reflected in the IBKP portal in terms of the first person who was authorized, because it was authorized to the institute. The person has submitted that application on behalf of the institute. Now the current portfolio in terms of what is actually functioning in the institute will be reflected in the IBKP portal. So you don't require a kind of a change in the IBKP portal itself in terms of the director who is now present at the institute. You can go back and modify that in terms of the IBSC, who is going to be the technical person who is not as the IBSC chairman, but the institute responsibility can be changed in the ongoing pro forma that you usually submit. You don't require a change in the original description, sir. It will not be done.
Okay, sir. Okay. Sir, one more query, sir.
Yeah.
That risk group 1, 2, 3 pathogens,
Yes.
a less number of fish viral pathogen has been mentioned.
Okay.
So, for example, herpes virus. So there is a many number of herpes virus are available, but when we are seeing the list in the class 2, herpes virus of reptiles, herpes virus of mollusk and crustaceans was listed, but not herpes virus of fish. So there is a serious pathogen is available in India, present in India on cyprianus herpes virus. So in that case, if I want to work on vaccines of cyprianus herpes virus, so what is the procedure, sir? How we have to go further?
Yeah. So one thing to be added here in terms of a requirement is the list is only a defined list. It is not an exhaustive list in terms of what is actually present, what can be present. Okay. So it is always the RCGM always welcomes proposal in terms of inclusion of new items to the list of infectious organisms from the IBSC with supportive evidence. It will add on, because like the revision of the list gets updated every new addition comes in. So if it's going to be a scenario where you want to add something, put it to the IBSC and then forward it to RCGM for inclusion based on what scientific evidence you have. Second, in terms of a pathogen, which is going to be something already existing, it is not something which is going to be new to the country, which is already existing but not defined in the list. If you really feel that it comes under the category of risk group 3, because like what happens is when you put the applications to RCGM, it doesn't look into the guidelines which is already existing into the country. It also looks into the guidelines of other countries too, in terms of European Union, in terms of FDA. It looks into all the guidelines, because as a country nominated to ICH guidelines, we look at harmonizing things. So it will always look at when you put a new pathogen and say that this is a kind of a serious pathogen, I want to work on it. It will look at other existing list in terms of other countries and goes back and revises that. That is one which revision can be done by putting an application. If the pathogen requires handling in terms of a biosafety level 2, if you put up with appropriate justification in terms of the containment, in terms of disposal, and what is actually required abroad, if people have already defined, you can get that approved in terms of what is actually defined by you. If it requires a biosafety level 3, it is not a scenario at all in terms of clearance. You just need to show where the work is going to be conducted in terms of the biosafety level 3 experiments. That's it.
So that application has been done by the respective IBSC or the person who is working on the pathogen.
The person, all the applications to the IBSC will be done only by the principal investigator.
Okay, sir. No, sir. For including the
Including what you can do is like you can add that to the other items in the IBSC minutes. The individual can put it to the IBSC, and the IBSC chairman can forward that on behalf of the IBSC to RCGM. If you require any support in that, please let me know. I can add on the information to the appropriate authorities in RCGM to update the list.
Okay, sir. Thank you. Thank you very much, sir. I'll be in touch with you for this purpose.
Sure.
Thank you, sir.
Any other questions, students?
Thank you for the
Thank
option,
you.
sir.
Yes. Thank you, sir. It's a really wonderful talk, almost an hour. Yeah, more than. Sorry for that. No, it was very informative and very thought provoking. You are DC involved, sir. That is why you passed. No, it shows the experience and